For those of you that are joining us, good afternoon, everyone. We will be beginning our program in just a couple minutes. Hang tight, and we'll be right back. Welcome, and thank you for joining us for Ask Me, our educational webinar series. My name is Steve Goklowski, Vice President of Care Services. I want to thank our presenting sponsor, ALS BioNews Today, and our accessibility and inclusivity sponsor, Shionogi, as well as our mission benefactor families and everyone who helps support our programming. Programs like this one, as well as the services we provide, are made possible through these generous donations. Now and always, we want to ensure you know that the ALS Network offers a menu of services free of charge to people with ALS and their families. Please know that if you need a piece of equipment loaned to you, emotional support or counseling, assistance with finding home care, help understanding insurance benefits, guidance in finding an ALS specialist or care, or a clinical trial, or just someone with expertise to answer any other questions about the disease, the ALS Network is here for you. Our committed staff, who are locally based in California and Hawaii, provide high-quality, personalized, and local services. As we also fight on the legislative advocacy front and build our growing research program. We are your local team, and we're glad to provide multi-layered support. To learn more about ALS Network and programs and services we provide, please visit www.alsnetwork.org for more information. Today's program will be co-hosted by Dr. Sarah Doherty, the ALS Network's Senior Director of Research. I'll now hand things over to Dr. Doherty, who will introduce our special guest speaker. Take it away, Sarah.
Thanks, Steve. Good afternoon, everyone. Thank you all for joining us. Today's program provides an overview of outcomes from the 2026 Packard Center Symposium. At the ALS Network, our research program is designed not only to fund innovative science, but also to actively engage with and contribute to the broader ALS research community. We see real value in showing up, participating in meetings such as the Packard Center Symposium, listening, learning, and helping to connect ideas and people across the field. These moments of shared insight are critical to strengthening the overall research ecosystem and accelerating progress. We hope all of you benefit today from that mission. I'm pleased to introduce Dr. Christine van de Velde, who is a full professor in the Department of Neuroscience at the University of Montreal and the University of Montreal Hospital Research Center, as well as the scientific director of the Packard Center for ALS Research at Johns Hopkins. Her research interests are centered on understanding the underlying pathological mechanisms that lead to ALS and FTD. We are thrilled to have her with us over the past 2 years. Thank you for being a returning guest in providing these important research updates. Thank you for being here, Dr. van de Velde. And with that, I will pass it over to you.
Hi, Sarah. Thank you so much. Thank you for the warm invitation once again to join you guys and share what has been going on in the Packard ALS research community. So I've thrown some slides up. Hopefully they're good.
They are good.
Wonderful. All right. So thank you to everybody on the call for joining. I am super thrilled to share with you what was the outcome of the 2026 meeting, which was held in Baltimore just last month, actually, a little more than 4 or 5 weeks ago. Before we jump into kind of the highlights of the meeting, I thought it'd probably be useful just to frame what is the Packard Center. Maybe not everybody is familiar with it. It is an ALS research center / international consortium based out of Johns Hopkins that is now in its 26th year. The overlying vision and mission statements are up on the screen. Really, our primary focus is really about getting toward. I got to move this to another screen. I feel like I need to do this. Is about really about how to empower collaborative and breakthrough research. This is the #1 thing for the Packard Center, and we do that in a number of different ways. One of which is engaging the global research community through the symposium that we hold each year. Underlying the Packard, really all of the research projects and teams that are associated with the Packard, we really have one mission, and that one mission is to try to identify disease-modifying treatments and valid biomarkers that give us a deeper understanding of the disease. And so this is an image from a colleague of mine up here in Montreal, Heather Durham, which she had an image here of this beautiful, healthy neuron that's taken from a human ALS spinal cord. And we want to understand what are the processes that will give rise to these very sick, dystrophic-looking neurons that are not functioning well and are ultimately the ones that are affected in ALS. And so we want to understand this process so that we can stop it, treat it, and effectively halt ALS. So when we think about the research ecosystem, there are many different organizations implicated, and all of them are fundamentally important to, I think, what is our shared mission, which is to improve the lives of ALS patients and really end up with a situation where ALS is not what we know it to be right now. Packard Center's spot is really of feeding the pipeline. And so this is an old cartoon that highlights sort of kind of these various steps that research goes through in order to arrive at the final coveted FDA approval of a drug that really shows efficacy in patients. And so before the clinical trials is all of the work before with identifying what are particular mechanisms or targets that could be developed or exploited for therapeutic development, screening and improving upon the molecule or compounds that have been identified, and then optimizing the chemistry. So Packard is really here at the beginning, at the very, very beginning, is understanding what are the targets, what are the biology and the mechanisms that go into these, leading to motor neuron loss, and how to prevent that. So this is where Packard's primary focus has been, and this is also in part and parcel with this is biomarker development, because a good therapeutic is, we only know if it's really a good therapeutic is if we've got good objective biomarkers that are also demonstrating a consistent effect on the disease. So biomarker development, if we think about neurofilament L, for example, and now some of the TP43 derived cryptic peptides that would fit in here. So I really see Packard as, I like this cartoon because Packard is really this, it's a catalyst. It's a catalyst in funding and kicking off projects, and as well as creating a community. And we do that monthly with our monthly PI meetings that are for funded investigators to share unpublished results and resources and basically accelerate the pace of research. We understand the urgency that families feel. We take that to heart. And so we really we do rapid sharing of data and resources in a monthly Zoom format that's just for researchers. And then we also host this annual meeting that I'm going to tell you a little bit about. in which we have a significant number of people in the room that all want the same thing. So let's talk about what the meeting is. So each year, the Packard for 26 years has hosted an annual meeting in Baltimore. This year's iteration included 327 participants, which included leaders both from the academic research environment as well as industry leaders. So coming from large pharma, also from smaller biotechs, as well as representatives from federal agencies and a number of nonprofits where we all share the same goal. We're also in the room. We also, in addition to the 327 people that we squeezed into this one room, we also had another 125 people that were joining us on average each day to listen to what culminated in 42 research talks. These are rapid fire research talks, 42 speakers, one amazing keynote speaker that I'm going to tell you a little bit about. We also had something called a poster blitz session in which young investigators, so students and postdocs, so those are the people that are actually in the lab doing the work, they gave us 3-minute presentations about their work, what their work was, and that was followed up then with 2 evenings of folks visiting their posters or their big 4x8 posters that are sitting up on the wall that people go and look at. We had 100 of those. This was a record number this year. I think Susie Connolly, my colleague at Packard, who helped organize this meeting, figured out how to put 100 posters in the space. We also coordinated networking lunches in which these young investigators can meet with leaders in the field from different sectors that are looking for how to develop their career and make contacts. There was also a panel on artificial intelligence and how this can help our research. And we had 21 partner organizations in the room. This was really an unprecedented turnout for this meeting. The emphasis on this year was on network and collaboration, and we're really grateful for our partners that came out. And for the research was phenomenal. It was primarily unpublished work, which is the Packard tradition, and is actually what makes the Packard meeting stand out over other meetings, is this very, very strong emphasis on sharing early research prior to going through the publication process. Again, focusing on the urgency of the research and making sure that we get things moving as fast as possible towards the clinic. So the theme, no surprise this year, was around collaboration and how we take that term to heart, both in the lab and in the organization and amongst organizations. And if you'll see in the background, there is also hope underlies all of that. And so one of the taglines of Packard is the hope is in the science. So with that, we're going to switch a little bit to science. And I want to highlight that we have this year launched a new program called Packard Partnerships. And this is just a sampling of some of the various organizations that we've worked with co-funding some of the research that you're going to hear. We are grateful to all of them. And I'm going to highlight ALS Network because I'm with you guys. Thank you very much for co-funding a grant and allowing me this opportunity to share what's been going on in the Packard research community. So the meeting this year was opened by Ed Rapp, who is an individual that is also living with ALS and an advocate for the community at large. He is actually the chair of the Answer ALS Advisory Board, which is a large consortium of patient-derived iPSCs, or induced pluripotent stem cells, a resource that researchers can access to generate motor neurons that we can study the neurons that are affected in patients. And he's also been a member of the Packard Board of Governors. And Ed, more than anybody, really embodies this idea of collaboration. And he gave a fantastic talk for the room, really grounding the conversation around collaboration and how we are each a domino. And I don't have my domino. He gave us each a domino, which is sitting on my desk at home, in which we talked about how you just need one domino to fall in order to put all the pieces in place to solve this disease. And so I just want to highlight, Ed gave a fantastic talk. If you have never heard Ed talk, I highly recommend it. He understands the science well and really speaks to the collaboration, and we're grateful to him for that. The meeting was kicked off this year with, after following Ed, the science component was kicked off by our keynote address from Sergio Pasca from Stanford University. Sergio is a leader in the field where it comes to thinking about organoids, which is, I'm sure that's probably on the call, I've kind of heard of little brains in a dish, or mini brains, is often called in the contemporary press. So Sergio, Dr. Pasca has developed various models to study how spinal cord and neurons and the surrounding cells that support them and the brain, and trying to figure out how to model them in the dish. She showed us some beautiful images, just absolutely beautiful microscopy, which I don't have here. Those were on his talk. It's showing us how it is possible to assemble sort of a little mini nervous system within a dish, and this allows us to understand better how the neurons in our brain connect to those that are in our spinal cord, which then connect to the muscles that drive our voluntary movements. And so he's created these beautiful structures that has provided significant insights to how we can understand how these denervation processes happen in the context of disease, and then also how do we leverage that to identify potential therapeutics that could stabilize these connections. He gave a fantastic talk, and the room was very much in awe of the work that he showed us. So really, it was a beautiful kickoff to the meeting. And choosing Dr. Pasca to be the keynote was strategic in a way, because there were some themes that had started to emerge as we crafted the program. And so that's the highlights I'm going to give you here. We're going to talk about the focus on neuronal firing, and then we're going to talk about looking beyond neurons. So often we hear in ALS always about the motor neurons, but what about the other cell types that live within the nervous system, and how do they help support those neurons that are lost, and what kind of things can we do to start thinking about them and their biology. And then a section that I call new and exciting, just cool stuff that I'm really excited to share with the community. Okay, so the focus on neuronal firing, and so why am I making a focus here? So neurons are electrical cells, and they transmit electrical impulses between each other in order to contract a muscle, right? To move your hand, to move your toes. It's a firing event, and so this idea of neuronal firing, it's a communication mechanism. It's how the neurons communicate to each other, and it's by sending a signal, and we call it firing. And we know that there are the neurons, the motor neurons, that are responsible for controlling movement, but those motor neurons themselves are subject to regulation by other types of neurons. And so there are neurons that can excite those motor neurons, so they drive them to fire more. And then there are those neurons that are inhibitory, which actually inhibit the firing. And so if you think about how you extend your arm, right, you've got to activate one set of muscles, your biceps, your triceps and your biceps, it's coordinated muscle contraction. So, and that happens by this combination of firing on the motor neurons and this action of excitatory and inhibitory neurons on those motor neurons. And so this idea that there was perhaps an imbalance of excitation and inhibition that contribute to motor neuron demise was something that emerged in this meeting. It's been around in the field for a very long time, but I think that it was something that was very much on display this year. And so one of the first talks I'm going to highlight, so I'm going to give you a series of vignettes of, I think, about 9 different talks out of the 42. So you can see I'm really only scratching the surface of what was covered by the community. But this one here is from Monica Gorosini from the University of Alberta. And so full disclosure, I am Canadian. And then so Monica, Dr. Gorosini, she showed this very interesting new approach that she has, a new mechanism that she's uncovered that she thinks might be a useful biomarker. And so I want to draw your attention to this little cartoon up here at the top. This idea that in ALS, there is a pre-symptomatic phase and then there's a clinically manifested phase. And that somewhere along the line, we have something called phenoconversion, so right, where an individual converts from being asymptomatic to being symptomatic. And this has been, there's been extensive studies about how neurofilament levels rise and that during this process, and that this is indicative of axonal damage that's accumulating. But this happens maybe kind of late, right? Because if neurofilaments have been released, that means that the neuron has been damaged. And so it would be nice to have something that is much, much earlier in the process that would help indicate, for example, in an individual that is a gene carrier, whether they should be provided treatment or not. And with this, I'm talking about SOD1, for example, with Qalsody. And so what Dr. Gorosini showed was that there was an electrophysiological signature that she could pick up in the muscles of patients. And so Dr. Gorosini actually comes from the spinal cord injury field. And so this is what inspired her work to look to see whether or not the signature that she'd identified in spinal cord injury patients, whether that might also be detected in ALS patients in their very earliest symptomatic phases. And the answer was yes. It's something called persistent inward currents. And so this idea that ions need to move back and forth across the neuronal membrane in order to get them to fire or to transmit signal. And so she identified a particular type of signature, an electric physiological signature that looked very promising and could potentially be a potential phenoconversion biomarker. And so her talk stimulated some discussion around the room from various people there, clinicians, scientists, and those involved in clinical trial development about whether or not this might be something that could be useful and implemented in current ongoing trials. And so I'll also just mention that at Packard meetings, we have 2 microphones in the room, and there's usually no less than 5 people lined up for each of these microphones at any given time. So there was a lot of great discussion around this particular presentation, and we look forward to seeing how Dr. Gorasini's discoveries on these persistent inward currents might be implemented in a clinical setting. And so I think more work to come. But I can say that I think that she made some very strong connections with various clinicians that are focused on ALS in the room. The second speaker I'm going to share is from Dr. Evangelos Kiskinis at Northwestern University. And so Evangelos is somebody who studies motor neurons that have been derived from iPSCs, which I mentioned earlier, so induced pluripotent stem cells. This is the idea that you can take a skin cell or a blood sample and you can reprogram those cells to be what we call pluripotent, meaning that they're in a naive state that you could, with the right sort of ingredients, you can trigger them to differentiate into any cell type of the body. And of course, in our context here, we're interested in motor neurons. And so Evangelos, Dr. Kiskinis was one of the original investigators that worked on iPSC motor neuron models for ALS. And in his most recent work, he has been digging deeper into what other types of neurons might be participating in the disease. And so what the experiment that he looked at was looking at neurons that have TDP43 pathology or don't. And I'm sure many on the call have heard a lot about TDP43, an RNA binding protein that normally lives within the nucleus, which is the command center of the motor neuron. But in the context of ALS, more often we see it outside of that command center. And so we see it out in the periphery. And so trying to understand what the consequences of that are. And so what Dr. Kiskinis does is that he took human postmortem material that's been very generously provided by patients and separated out those neurons that had TDP43 still in the nucleus and to those neurons that didn't have it in the nucleus. And then he asked, what type of neurons are they? And what he discovered was that the ones that have lost their nuclear pool of TDP43 are almost exclusively excitatory glutamatergic neurons. And so that means is that in this idea of balance of excitation and inhibition, which I touched on the previous slide, is that if we've lost these excitation, there's dysfunction here in these excitatory neurons, and now this might contribute to why the neurons don't fire in the proper way that they should. And so, by extension, he also identified that the inhibitory neurons, so the ones that put the brakes on the motor neurons, these were not the ones that were affected at all by the loss of TDP43. It was really just the ones that are activating or excitatory. So that's very, very interesting because these are the ones, so motor neurons receive glutamate signals. And so these excitatory neurons are the ones that are secreting glutamate. That's the neurotransmitter that is being activated that acts on those motor neurons. It was a very interesting study in which he really isolated what type of neurons were being primarily affected, which had not actually been robustly studied prior to that. So now that we know a little bit more about what particular types of neurons that are affected, now we can sort of start to target our approaches to understand the biology in those neurons, because not all neurons are affected in the same way in ALS. And so trying to understand where the differences is key to understanding developing therapeutics and biomarkers. So the next section is going to be about looking beyond the neurons. And so this is the idea that neuronal death is non-cell autonomous. And so the term non-cell autonomous refers to the idea that other cell types also contribute to the disease process. And so what are those other cell types? And so I'm going to do a quick little lesson here on the different cell types that we most often speak about in the lab, in the research community. So I'm sure most people on the call, we always hear about the motor neurons. This is the motor neuron with the very, very long axons that project all the way from your lower back all the way to your big toe to innervate your toes. But also they project from the upper cortex onto the ones in the spinal cord. These are the ones that we talk about all the time, but there are other cell types that are equally important to the well-being of these very, very valuable motor neurons. So one cell type is called astrocytes. And so astrocytes actually help to maintain a very healthy environment around the neuron. They clear away kind of toxic they clear away the extra stuff that the extracellular, it's called extracellular glutamate. So that extra glutamate that's sitting there, that's making the neuron fire. The astrocytes are tasked with they're like sponges, and they suck it all up to get it away from the neuron, because the neuron can't handle this continuous neurotransmission with the glutamate. They also provide lots of trophic factors to support the health of these neurons. And so they're very, very important cells. Similar to those are the microglia, which are kind of like the immune cells within the brain and the spinal cord. They kind of are always patrolling, and they're looking for making sure the environment is nice and clean and doesn't have any pathogens or aggregates sitting out there. And so it's kind of like the vacuum cleaners of the nervous system, and they support the motor neuron as well, because the motor neuron doesn't want to get jammed up with a bunch of junk in it. And so the motor neurons eject it, the microglia chew it up. And then the other cell type of interest is also called oligodendrocytes, which these are the ones that wrap the very, very long axon. They wrap them with myelin. And the myelin is really important because within the central nervous system, these are the ones that help the neurotransmission. And so it's really important that these oligodendrocytes are very, very healthy, and they help the neurons to fire. And a neuron that's firing properly is a happy neuron. A neuron that is not firing correctly is stressed, and it's not going well. So this concept is that each of these cell types actually contribute to the health of the motor neuron itself. Okay, so now with that lesson in mind, I'm going to share with you work from Dr. Rita Sattler at the Barrow Neurological Institute in Phoenix. So Dr. Sattler has been investigating a novel protein that's expressed in the glial cells. So that's in the microglia, the astrocytes, and the oligodendrocytes. Those are collectively called glial cells. And she's identified this factor called SPARKLE1, which is expressed at the synapse, so where the neurons make contact with each other. And so it's thought to be expressed at the astrocytes and helps anchor and support that synapse. So when you have a synapse coming in together into neurons, you have the astrocytes that are kind of the braces around it that help stabilize it and keep the synapse in good health. And so she's identified, using, again, patient material, this time from patients that have the C9 expansion. She's identified that this molecule called SPARKLE1 that is dysregulated in the C9 patients, and so it seems to be downregulated. And so she's been exploring using IPS derived cultures of motor neurons and astrocytes together, where she's co-cultured them together in a dish. What is the consequences of not having enough SPARKLE1? And so what she's basically found is that there seems to be a loss of dendrites or synapses. We'll stay with that word. And that seems that this molecule is really truly helping to stabilize those structures, and that this might be something that is relevant to disease, since it has been observed previously that synapses seem to be lost in clinically silent stages of disease. And so this concept of synapse loss is something that's been appreciated in the Alzheimer's space for several years, and has been floating in the background for a while in ALS, and is now coming to the forefront. So that was very cool, exciting work. This is a funded project with Packard. I'm also going to tell you about Dr. Jonathan Kupernock from the University of Sheffield. And so Dr. Kupernock is a clinician scientist and with a very strong focus on genetics. And so he has, with a collaborator, Sai Zhang now from Yale University, has leveraged large-scale genomic data sets and machine learning tools to identify a new ALS gene. And the new ALS gene is called WDR49. And what's interesting about WDR49 is that it's really primarily expressed in astrocytes, those support cells of the neurons. And he actually shows that this WDR49 expression, which here are this red and yellow labeling here, provides neuroprotection. And so astrocytes that are expressing this molecule actually are actively supporting a neuron. What was really cool, probably the coolest thing in his entire talk, was that he found that those astrocytes that were expressing this new molecule, WDR49, were actually close to neurons that were showing TDP43 pathology. And so this idea that there's cytoplasmic TDP43, that RNA binding protein that I mentioned, it can be sitting out in the cytoplasm. And so you found ththose WDR49 astrocytes close to neurons that were basically signaling that something is not right. And so the question is, is there some sort of signal for help from those neurons that are showing or experiencing TP43 pathology, that then the astrocytes come in that have this neuroprotective factor that then try to help to stabilize and nurture that neuron. And so that was very, very interesting. And this all came out of, again, leveraging a very large data set of ALS patients from around the world. And it is the first one that I think that is actually a gene that is really known to be primarily functioning in astrocytes. As we know, in ALS, there are more than 40 different genes that have been associated, and I think several of them are expressed everywhere. But this one really seemed to have particular enrichment within these astrocytes, which is very, very interesting. And again, speaks to the idea that it's not just the motor neurons that are implicated in the disease. The other cell types close by to the neurons are just as important. The next little vignette is from Dr. Jimena Anderson from Emory University. She is a young, new investigator that is actually just coming to the ALS space. And actually, I want to highlight Dr. Anderson because the keynote speaker that I told you about, Dr. Pasca, she actually trained with Dr. Pasca. And so those models then which I mentioned about how to assemble sort of cortex and spinal cord and muscle assembloids in a dish, it's actually Dr. Anderson's work during her time with Dr. Pasca. And so she has just been funded as a new investigator with Packard and Live Like Glue, in which she is actually working on a project to develop organoids using iPSCs to develop organoids that incorporate these oligodendrocytes. And so it's a very, very exciting new project. This has been a cell type that has been traditionally extremely difficult to culture and model in a dish. There's a lot known about oligodendrocytes in general, but mostly coming from mouse-based studies. And so this idea that we now could have a human-based model is very, very exciting, as they're essential to supporting neurons and enabling the neurons to fire normally. Again, it speaks directly to non-cell autonomy, and we know that this cell type actually can contribute to disease because a long time ago, in mouse models that were using the SOD1 transgene, we actually showed that the field showed that this cell type is affected in ALS, and actually early dysfunction of the oligodendrocytes might be part of the early phase of the disease. So we're very excited to be supporting her and to hear what her plans were and exploiting her technology with these assembloids. Here's an example of this. This is the cortex, spinal cord, and muscle, just to see how to exploit that to study oligodendrocytes. So more work to come. We're going to hear from her in the coming years. Again, an exciting new area. Another project that I'm also quite excited about is from Aaron Burberry at Case Western University in Cleveland. And so Aaron, again, is also a young investigator who comes from, a long time ago, he comes from one of the groups that also pioneered the use of induced pluripotent stem cells to model motor neurons. And part of what he's shifted focus a little bit now, but looking more at what is the relationship between environmental factors and ALS? And so we hear often the questions about environmental factors, and with which there's really a poor consensus in the field about what those are. But here he's leveraging the idea of being able to manipulate the diet. And so this gut microbiome access is something that has been in the popular press in many neurodegenerative diseases in recent years. And it's the idea that maybe kind of, the not that you are what you eat, but it's the microbiomes, the microorganisms that live within your colon that, basically help us survive, that they might be processing food in a way that is maybe not beneficial and can maybe activate inflammatory processes that can be irritants to the neurons and the nervous system. And so he identified that there's a high production of something called glycogen. It's a special type of sugar that is produced by bacteria in the gut, and that this particular type of sugar actually could stimulate the immune system. And so he demonstrated that using patient samples. And what was interesting is that he found that the C9 protein, the protein encoded by the C9orf72 gene, actually blunts this response. And so that C9orf72 protein has been implicated in immune responses that's been demonstrated previously in mice. This was a beautiful demonstration of showing this in humans using human samples as well. So a very interesting study in which this idea that inflammatory forms of the glycogens might be more prevalent in the guts of individuals affected with ALS or FTD. That was also included in his study. And he confirmed this in mouse models where he treated mice with this particular sort of enzyme as a proof of principle that if you got rid of the glycogen, that you could improve the profile of these mice. So it's an interesting idea that is now being explored further by Dr. Burbury, this idea of how do we, is diet modification something that really could be something accessible as a potential treatment option for patients. Very early days, but it's very, very promising work. What about new and exciting? So this was us in the audience when we heard some of these next stories. Very, very excited about the following few stories. And so the first one I'm going to present is a collaboration between Sami Armata and Eran Hornstein. So Dr. Armata is a clinician scientist at the University of Michigan, and Dr. Hornstein is a scientist at the Weizmann Institute who has developed a novel approach to imaging cells in which, using a machine learning approach, he actually can image about 30 different subcellular compartments in a cell at the same time. And so it allows you to shine a much larger light on what's all happening when for example, TDP-43 is not in the nucleus. And so he's coupled this with Dr. Barmata, who actually has developed a system in which TDP-43, which is normally in the nucleus, can be forced out into the cytoplasm, so outside of the command center of cells. And then you can switch, and do this with light. And you can switch the light on and off, and you see that within just a matter of minutes that the TDP-43, the protein, can go back in the nucleus, out the nucleus and in. So you can cycle it in and out. So this is a very, very exciting model or cell culture model, because it's really difficult to model TDP-43 mislocalization events that we see in postmortem material. It's really difficult to model that using any other approach, actually. And so now what you can imagine is that if you were to pair these two things, that you had a cell in which you can switch the TDP-43 localization just by flipping the switch on the microscope. Now you could study the consequences in a more holistic way on 30 different cellular organelles at the same time. And so maybe that, because right now I think what we have is that we see what we look for. And so here, this is a much larger lens that we can look to see what are the consequences of TDP-43 moving in and out of the nucleus, with, again, reminding everyone on the call that TDP-43 mislocalization or translocation to the cytoplasm is one of the most common histopathological features in ALS and FTD. Next, I'm going to tell you about Dr. John Landers at UMass Medical in Worcester, Massachusetts, Dr. Landers is a geneticist who has been contributed to many of the key genetic findings over the years. He has taken his expertise now to develop something called allele-specific antisense oligonucleotides. And so for those of you on the call that have heard about antisense oligonucleotides, that's probably been in the context of tofersen, or now known as Qalsody, which is an antisense oligonucleotide that lowers the expression of SOD1, but it does that in a way that it's not mutation specific. It lowers all the SOD1. But that's okay, because the research has been done to demonstrate that it's okay to lower the human SOD1 protein in human neurons without too much consequence. In fact, it's beneficial. It's okay, because you get rid of the mutant allele, but you also get rid of the wild type, but that's okay. But for some genes, you're not going to want to get rid of both the bad allele, the mutant carrying allele gene, copy of the gene, and the wild type. Sometimes you need to keep the wild type because you need the function that it's doing. And so one example of that would be TDP-43 itself. We know that TDP-43 is essential to neuronal survival, so we would not want to do things in which we would be knocking down all of TDP-43, both the good and the bad. We want to develop something that where you can just knock down the mutant. And so Dr. Landers is developing a method in order to do a mutant-specific knockdown, so ensures the normal expression of the good copy of the gene. And this leverages genomics because he's identified small sequence variations that are sort of inherited along with the mutation, that he can then target antisense oligonucleotides to those regions. So they're not targeting the mutation directly, but a region that is somewhere downstream or upstream, so that he can then target more than one mutation at a time. And so if you think about all the energy that goes into developing an antisense oligonucleotide, there's a lot of characterization and a lot of research goes into getting one of these into trial. And so if you had to do one for each different patient, it could take a lot longer versus this is my little cartoon that I had ChatGPT help me draw. The current approach is one stone, one bird, but imagine if one ASO could hit 5, 10, 15 different mutations, and how much more it could be effective for many more patients, and then would actually shorten the time required to get to an individual. So this is very exciting, very early work, but very, very exciting to see how this is evolving. He had great proof of principle using iPS-derived neurons in a dish, and so the next steps are coming. And then I think the last one I wanted to share is from Dr. Michael Benatar at the University of Miami. And so Dr. Benatar is well known in the ALS research community for his efforts on what's something called the pre-FAL study, in which he's been following SOD1 gene carriers over the span of many, many years. And he is really looking for, this is a cartoon I showed previously, but his work has been focused for many years on trying to anticipate when someone is going to convert from a pre-symptomatic to a symptomatic individual. And so, again, I highlighted about neurofilament, but what Dr. Benatar here has done is he's leveraged blood samples from several patients that he's followed longitudinally over many, 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 many months, many years in some cases, and trying to identify, is there a panel of proteins that might be in the blood, for example, that might be informative of when a patient is about to convert from an asymptomatic to a symptomatic state. And so he's actually identified a panel of 19 proteins that he's tested now on a small subset of patients. And really his goal is to try to predict that phenoconversion. When should we treat? And so this is very relevant right now for the SOD1 gene carrier population. Is there, at what point should somebody receive Qalsody? This is a very, very salient question in the field that is going to also be relevant to other genetic subsets in the community. And so Dr. Benatar is leading this work. Very, very exciting to see how this is going to evolve. So the key takeaways here is that the research community at large in ALS is very highly collaborative, and the Packard fuels that by convening this international meeting each year. We saw a lot of new cellular and molecular targets, and excitingly this year, electrophysiological targets. Exciting because these are things that can be actioned in the clinic. So we'll see what's going to come in the next year. And remember when I talked about posters and poster blitz? So this is a nice, put this photo in to show. This is what these posters look like. And these are examples of young students or postdocs, individuals working in our labs that are actually doing the work. And this is how they put all the stuff up on a big, big poster, exactly as I mentioned. And then we spend time talking about them for several hours. There's 100 of them like this year distributed around the space. It was very crowded, but totally worth it. It was fantastic. And so just so you see some of the what it looks like, this room is packed, completely packed. There's rarely an empty seat found in there. This person's probably just gone to the bathroom. It's always very, very full. We had fantastic talks from Dr. Maragakis at Johns Hopkins, Dr. Jinsy Andrews from Columbia. This is Dr. Pasca, who I mentioned earlier. And this is Dr. Daniela Zarnescu from University of Pennsylvania. So I hope that that gave you an idea. If you want more information, feel free to reach out to packardcenter.org. The email is here. And again, the Packard Center is about collaboration, and we're excited to be collaborating with the ALS Network, as well as all the other organizations that are in the space. So thank you, and I'm happy to answer any questions.
Thank you so much. That was such a fantastic rundown. As someone who was in that crowded room, it was a phenomenal meeting. You are not wrong in that every time that a talk was given, there were folks lined up at those two microphones to ask questions and to talk. And it was so exciting to see in real time ideas being shared. And have you thought about trying this or that? And it's a wonderful opportunity. So that was, you do not oversell the amount of discussion and ideas that are really being shared in that room and at the poster sessions. Yes, it was an incredible, incredible meeting. So thank you for giving us that rundown. I will start off. We have a question here regarding treatments on the horizon. So starting off kind of broad, are there promising treatments or therapies being developed to cure ALS? And what about specifically halting the progression of sporadic ALS? So kind of zooming in on that sporadic.
Yeah, so I think there are a number of things that are coming forward. So I think the one that has got a lot of buzz in the field right now is Stathmin-2 antisense oligonucleotides from Curalis. So Stathmin-2, so I remember I mentioned about TDP-43 and the idea that it moves in and out of the nucleus. And one of the things that TDP-43 does is to control how genes get spliced together. And so when TDP-43 is not in the nucleus like it should be, there are lots of different genes that are affected as a result. Stathmin-2 is one of them. And it's an important one because it's required for, it seems to be required for maintaining the axon, that very, very long extension of the motor neuron. So antisense oligonucleotides have been developed in order to prevent the splicing event that inadvertently happens when TDP-43 exits the nucleus. So it's not like Qalsody, which is degrading the SOD1 RNA. This one is actually trying to preserve the Stathmin-2 RNA so that the Stathmin-2 levels stay up. And so a phase 2 was just completed, or pardon me, phase 1, I believe, was just completed. And the company is now rallying for phase 2. It's hard to say, there was a press release put out about it. It looked, there weren't negatives. There were not so many negative words used in the press release. And there's definitely been there must be enough positive signal there that they are, at least, don't forget, phase 1 are really for safety and tolerability. It's not about efficacy. And so they are now gearing up to do that phase 2 study that will give us insights about efficacy and therapeutic potential.
And
so
can you
that
remind me, is that one being run in the US or is that one in Canada?
I believe it's being run in Canada. You're correct.
Okay.
Yes. Sorry.
That's all right.
You're right. Yeah, it's being run in Canada. There's also another one from Corcept, which is a glucocorticoid receptor. And I actually saw this one at your meeting, actually, in Pasadena. There were several posters from Corcept that I thought were really interesting. So this one was, they're gearing up to do a phase 3. And so their phase 2 study was something like just over 200 patients. And there, what they showed, so I know that most clinical trials rely on ALS-FRS as a readout. This one didn't. In fact, it seemed that the signal comes from survival as an endpoint, which is a little unconventional because we typically moved away from using that as an endpoint. But this idea of stress, so psychological stress, so that's the glucocorticoid receptor is activated by psychological stress. And so this idea that blocking that might be beneficial. And I find that very interesting because I think we don't understand fully the effect of stress on the body. And so I think that one is going to a phase 3 soon. And I'm excited to see how that looks. Some of the mouse models that they showed using, again, not necessarily conventional mouse models. They're using the wobbler mouse, but showed something that looked kind of promising. And then there's also one called Prolinia, which is a sigma-1 receptor blocker. And so sigma-1 receptor is one of those rare ALS genes. And so that is sigma-1 receptor facilitates the contact between two cellular components, the mitochondria, which are the powerhouse of the cells, and then the ER, the endoplasmic reticulum, which is sort of like, it's like your UPS office. It's the sorting center of the cell. And so how these two organelles intersect is governed in part by the sigma-1 receptor. And so Prolinia is actually gearing up to go to a phase 3 as well. It looks interesting. It's the biology of there is kind of a little bit known. The Stathmin-2 one is very well known. And I think that for the glucocorticoid receptor, we're not exactly sure, but you can't argue if there's really a change in survival and I don't care how we get there, we get there. So I think those are 3 that I would say are to be highlighted.
Yeah, that's outstanding. Thank you so much for that. This next question I think is a little bit related, but diving into custom bespoke medicine approaches. What progress, you touched on this a little bit, but just to make sure that we are answering folks' questions, what progress is being made toward more personalized or stratified approaches of treatment based on everything you know to date through ALS research? Is gene therapy or precision medicine like Qalsody the best pathway? That's a bit of an opinion question, but the best pathway forward towards a cure?
Yeah, so I think the closest one, I mean, so for the FUS gene carriers, there's J-Suffusin, which I now know got a new name recently, and I can't remember what it is, which is really for FUS patients. So that one, there was a case study that was published in the Lancet just a few months ago of 12 individuals that were FUS carriers that seemed to have it was not it's a case study, right? So it's not a proper clinical trial, but patients were all, it was all with compassionate use. And they all seem, several of them seem to show some sort of indication of benefit. And so I think that one looks very interesting. And I think that similar approach, so the lead behind that is Dr. Neil Snyder at Columbia. And he has launched a project, the ALS Silence or NLORM, with the NLORM Foundation, with the idea of being able to make these tailor-made antisense oligonucleotide approaches that target very specific gene mutations. And so those are ongoing, but those are all N of 1 studies, right? So each individual, they're tailored for the mutation of the individual, which is why we're excited about Dr. Lander's study, which then allows you to, for example, if you think of TDP43, there's about 50 different mutations reported in TDP43. So imagine, rather than making 50 different ASOs, right, which is a slow process and you have to tailor it for each individual, what if you could make 2 or 3 of them that you could cover all of them with just 2 or 3? Wouldn't that be cool? Right? So this idea, and that would just, again, that's the idea of accelerating the discovery towards the clinic. So I think that those are ongoing, and this is great for those individuals that have, a known genetic mutation. But as you know, the large percentage of patients, we don't actually know what the underlying genetic feature, or even if there is one, right? We don't know. And if is it just one gene, or is it 2, or is it 3, or is it 10 in combination? Unfortunately, when you think of I'm thinking of that fancy thing in Dan Brown's, the codex, and you have to turn the thing, and you gotta, unwittingly lined them all up and right?
Yep, absolutely.
Right. So we just don't know for a large proportion of the what we call sporadic patients. We just don't know.
And that actually touches on.
We don't have all the familials sold either.
Right. Well, and that touches on another question that came in, is that it feels like there hadn't been much movement on the sporadic front, and not a new FDA approved treatment recently. And I think that question came in, and I hear that a fair amount, sort of where is the progress? What big changes are coming up? And I think you already sort of hit on it, that it's a very fair question, and it comes up a lot, but ALS, and specifically sporadic ALS, is so biologically complex, and sort of hitting on all of those pieces with one sort of silver bullet at this point. We just simply haven't gotten there. The disease is so heterogeneous.
Yeah, but if I may, so I
Please.
Totally hear the sporadic ALS patient population, because I can kind of feel like maybe I can feel how they might feel that they're neglected. But let me tell you a story that is really cool. So there's a researcher at UCSF, Dr. Steve Altschuler, and Dr. Lenny Wu. They have got this really interesting idea, which is that maybe some individuals who have apparent sporadic ALS, that maybe they're sporadic, but maybe their disease, the underlying biology of their disease, looks like an SOD1 patient, or looks like a FUS patient, right? And so imagine, okay, it's still early. Imagine you could have an avatar for each individual. So they're using fibroblasts from the skin. They're culturing them in the dish, and they're trying to characterize them using machine learning. So this is where AI has entered the research space in a fulsome way. And they're trying to see whether or not they culture those cells from sporadic patients, and they culture, say, a bunch of cells from FUS patients. Do any of those look like those? Because now if you have a therapy for FUS patients, maybe you could then argue that therapy should be tried on that subset of patients that have apparent sporadic disease, but their biology looks like the FUS, the biology underlying the FUS mutations. And so that they have published about this idea that this machine learning approach could work. They're currently trying to do that for C9. And there's discussion, I think, underway about doing it for SOD1. This idea of, I've heard the story before, the debate before about whether or not QALSODY should be made available to all ALS patients. I don't agree with that. I think we have to understand the biology. And so maybe this approach that Dr. Altschuler and Dr. Wu is developing, maybe that makes sense, is that there might be a subset of patients that look like, that their biology looks like the SOD1 patients, and they might then be good candidates for QALSODY. So early days, but I think it's an unconventional kind of, it's a different, very forward thinking. This is a project that's also supported by the Packard Center. This is, I think, where the Packard kind of excels in that we can take those risks. This is a high risk, high reward project. It might yield nothing, but I'm a big baseball fan, and you miss 100% of those pitches that you don't swing at, right? So I think it's a swing that's worth taking.
Absolutely. I love it. I've been following that team for a while. I think it is an incredibly innovative, forward-thinking methodology. And like you said, if it helps sort of break down some of what feels like a barrier to treatment and some of the folks in a scientifically sound and rigorous way, it would be an incredible finding.
So, I mean, so we'll know. The project is rolling, and I hope to hear more about it next year at the Packard meeting. All Packard-funded investigators are obligated to share the updates on their project, and they have to provide progress reports. They're followed closely. That's another strength of the Packard, is the input. There's feedback, lots of feedback given from the scientific advisory board as well as from folks in the room. So stay tuned.
Excellent. And we do have a question about the meeting itself. Can folks living with ALS attend to the meeting, or is this specifically for the scientific community? I think you also mentioned that there's virtual options. So can you explain the availability of attendance for folks with lived experience?
Yeah, so it is for the research community. We have had some participants from the community. We usually try to, I'm not to say limit, because that's not the intent at all. The idea is to make sure that we keep the focus on the science. I very much enjoy attending meetings with families, and as we were talking in the pre-chat, I have been schooled by many ALS patient and their family about different aspects of the research that I was unfamiliar with. I appreciate that feedback so much, but the focus really is on the research. If somebody is really interested, I would say to reach out to Susie Connolly at the Packard so we can discuss. We have also had, as part of the NEALS, we've run a research ambassador program. And so this idea that folks that might be more, that have been sort of, it's kind of the NEALS research ambassador program, the Curly program, pardon me. If we call it Carly in Canada. The Curly program, this idea of making sure that those folks that are listening are sort of informed about research process, and that is, I think, an important consideration. So just reach out and we'll take it from there.
Excellent. I love that. We have a couple of questions that I can kind of group together. You mentioned the importance of understanding ALS disease mechanism, providing a target for therapies. Were there any discussions at this meeting about ALS triggering mechanisms? And I kind of bundle that with causal exposures. So roles of sort of getting at a causal lens.
So, well, this is one of the weaknesses in the field. Is that what are the causal events of ALS? What we don't know what they are. We still don't know what they are. We focus on the genetics because it gives us 100% like a way in that we can study biology. There's a very large study underway out of the University of Michigan right now, led by Dr. Stephen Gutman, who is looking at trying to characterize what he calls the exposome, the environmental exposure ohm. This idea of he does this through very, very detailed questionnaires with participants, trying to understand what their exposures might have been over a lifetime. It's really hard to pinpoint, but we're still interested in it. We realize that the environment is an important factor. In my So I also run a research lab at the University of Montreal up in Canada, and so it is actually an area that is near and dear to my heart. And we work with one of the mouse models that has a predisposing mutation in it, in which we actually expose them to some environmental stress to try to see if we can't precipitate disease and the underlying biology that has been reported using it from patients. And so as a way to try to model it, because none of these TDP-43 mouse models, none of them really recapitulate the whole disease sequelae in a way that's very satisfactory. Based on and this coming from, I worked on the SOD1 mouse for a long time, which has a very clear sequence of events that is very closely resembling what is observed in patients. And the TDP mice are a little bit weird in that they don't do it in the same way. And so we've been trying to add these environmental exposures to see if we can't precipitate that. So folks are working on it, but I'm using an environmental stress that is artificial. It's one that I'm allowed to do. The idea is exactly that: is how can we push the disease into manifest? And so and I'm not the only group doing that. There's a few other groups as well doing that.
And I think that's reassuring in a way as well. It is a gap in that we don't know the answer, but it is certainly something that is being actively investigated. So hopefully more answers soon. And I would not be surprised if, unlike other things in the ALS research space, that the answer might be multifaceted, that there might be multiple things or, combinations of things. And that's sort of part of, like you said, the puzzle of putting it all together. I realize that we are at time, but I'd like to just squeeze in, if you don't mind, one more
that's fine.
answer.
No
Let's end with some positivity and some hope. If you had to identify from this meeting and sort of conversations that maybe have happened since, where do you see the greatest momentum right now? What would you offer as sort of a big sense of hope that came out of the Packard meeting this year?
Oh, what a great question. Wow, that's a big one. What is the biggest thing, the sense of hope? I think it's the collaboration. Still always blows me away. I'm always impressed by the science. I mean, this room is full of absolutely brilliant people. I am extremely humbled to be in their presence and to be allowed to serve in this role for the community. Really, really amazingly smart people that freely share their ideas. Really a lot of exchanges. There's always clumps of people at the coffee break, and every year we get the complaint that the breaks are not long enough and we need more time to talk. And Susie and I, we've added another reception, and we've added more of this, and we've extended the lunch, and we're trying constantly to try to facilitate those conversations. I myself had a number of collaborators in the room that I was just not able to have those conversations as badly as I wanted, because I had this 2 hats to wear that day, which was unfortunate. I set up meetings with everybody after, that's okay. But this constant collaboration and the sharing of resources, people are really, really generous in the ALS community. Again, with that emphasis of, this is again another thing that's kind of seeded by Packard principles, is that sharing of unpublished data, but also of resources. And by resources, we mean cells, we mean mouse models, we mean antibodies, we mean any tool in the lab, that can help the research go faster. Those are really freely shared amongst the research teams in the room. And I think that's really exciting in the sense, it takes a village, right? It's going to take a village. And what I love is that the ALS community is really, really supportive of everyone. I saw a lot of nurturing of young investigators that are entering the space. That was also very, very cool. We had some very new investigators that are entering the space that they're sort of not sure, and you just see them get enveloped. And so one of them, for example, she's a funded investigator, and it was raised in the private SAB meeting after that, maybe this, it would be good to surround this investigator with a mentor. And so we did that. We provided that introduction. There was senior members of the SAB that put their hand up, that they said they would be really, really thrilled to help and to well frame this person and keep them in the community, and to help them to expand their research and what they might do. So things like that. I think that was really very, very exciting to see that.
Yeah, that's absolutely incredible. And I have to agree. I saw it in action as well. You nailed it. I think that the collaboration, the drive that is there in the community, it's
Oh, yeah.
incredible. That mentor program sounds like, or the possibility of a mentor program sounds incredible as well. So that's a fabulous idea. We are at time. I thank you so much for your time. I will call on, I think, Steve to come back up to wrap us up.
Right. Thank you so much. Thank you, Christine and Sarah, both for your time. Wonderful information. I want to thank all of you so much for joining us today. We are grateful to each of you for being a part of our community. And once again, a big thanks also to all of our sponsors for their support. We didn't get to all the questions, but if you do have other questions that were not answered today, including questions about the services offered at the ALS Network, please email us at askme@alsnetwork.org. The ALS Network brings together our community in various ways throughout the year to help create help and hope for families impacted by ALS today and for those who will be diagnosed in the future. These celebratory and meaningful gatherings provide opportunities for connection and raise awareness and funds to benefit the ALS Network's mission priorities. Please visit events.alsnetwork.org to learn how you can join us. As you know, philanthropy fuels everything we do, and we are grateful to the generous donors who make gifts of all sizes throughout the year, including contributions through an estate plan. To learn more about how you can support valuable programs like this one, please visit our website at give.alsnetwork.org. And please watch for the short survey on today's presentation that you will receive via email in order to help us plan for future programming. Thank you again, and we'll see you next time.
Thank you, everyone.
Thank you so much. Thank you, Dr. Vanaveldi. Have a nice day.
My pleasure. Take care.